discussion about the Ebola virus. And I am now giving you the lecture October 2014. For the past month or so, we've had an epidemic, so to speak, of Ebola virus in West Africa. And depending on when you actually listen to the lecture, it may be that many more things have developed since that time. Things are changing on a daily basis. And so what we're going to discuss today is what we know right now about the epidemic. We're going to discuss the structure of the virus, the influence and interaction with the immune system, the various precautions that can be taken in relationship to protection from infection, and some of the strategies that we're thinking about currently as of this time. So the outbreak distribution now, again, this is as of 1976. So you might say, why is that such an early date to think about? Well, that's the time that the Ebola virus was first identified. And at that time, there was a pretty sizable outbreak. And then there have been little outbreaks here and there since that time, but it's only really been the last month or two that the outbreak has really grown sizably to where it's being actually considered an epidemic. There's quite a lot of hysteria. There's a lot of fear. There's precautions being taken. The first patient to ever be diagnosed in the United States with Ebola just emerged a few days ago. And so it's becoming quite a big health concern, and the WHO has declared a public health emergency. The CDC in the United States has actually sent a contingent of 1,000+ health workers to really try and contain the epidemic in West Africa. So here you can see since 1976, but really the most recent outbreak epidemic of Ebola is occurring here in this part of West Africa with the Zaire Ebola virus, which is one of 4 different types. And that type in particular is the most common type, and that's been over time as well. So as of actually a week ago, right, since I'm giving this lecture today, you can see that the 3 countries most affected is Guinea, Sierra Leone, and Liberia. And the reddened areas are where the new activity is, and the orange is active, of course, and the yellow is so-called inactive. But really what's going on here is that it's, first of all, underreported. There may be many more cases that haven't been reported, people who've gotten the disease out in the outlying areas who, basically died and never received the count, so to speak. And so in that sense, there could even be a much greater epidemic that we don't even know about. But, you can see with these 3 countries, there really aren't that many hospitals in those countries. And Sierra Leone, for instance, only has 2 hospitals. Guinea only has 1. So, in terms of being able to treat all the individuals that are coming down with Ebola, it's become a huge challenge to these countries. And we're going to talk about that later on in terms of how to think about ways to eradicate the epidemic. But basically, the United States has sent personnel from the CDC to these areas to help the personnel there try and contain the epidemic. So let's talk about the lifestyle of the Ebola virus. Where does it live? How does it transmit? And so forth. So Ebola virus, way back in the early, or I guess in the late 70s, it was really a virus that was thought to just infect animals, mammals, of course, warm-blooded mammals, both bats and primates and other wildlife. And the problem becomes really when the virus actually infects a human, that the zoonotic infection of the Ebola virus infecting humans, that's where the epidemic starts, because once that happens, then humans can infect other humans. And in general, in microbiology, these zoonotic infections are generally very severe because the human organism has not had enough time to really evolve an effective immune response against these pathogens. So often a newly emergent virus or a viral infection that's transmitted by an animal is in the early times actually pretty severe. Another example is the HIV virus that is thought to come as a zoonotic infection from primates. So in any event, now we're experiencing this quite sizable outbreak epidemic, and we'll discuss in a minute how we think that actually happened. So the actual infection, let's talk about the infection actually coming from, or the virus coming from an animal, infects the human, and then we see that the actual cells in which the virus migrates to, the immune cells, the macrophages in particular, are the cells that become infected. And through this various processes, which we're going to talk about in terms of replication, more viruses are replicated inside the cell. Then they leave that macrophage and they infect other macrophages. But also in the inflammatory process of the immune system, if you can remember the immune lectures, one of the things that happens are the endothelial cells, which are the cells that line the blood vessels. Because of the inflammatory process, the actual gap between the endothelial cells actually becomes wider due to this inflammatory process. But then what happens is the actual gap becomes so wide that there's actual leakage of blood, which is one of the hallmark signs of the Ebola viral infection. And you can also get leakage of the virus particles or the virus itself. And so this is what we're actually seeing. These are some of the signs of the infection itself. So we'll come back to this slide at the end and we'll talk about it again in more detail. So the actual symptoms are, coming down with a fever. The incubation period is as short as 2 days and as long as 21 days. And what happens is the early signs are fever, headache, dizziness, vomiting, stomach pain, muscle pain, and unexplained bleeding or bruising. Now, these symptoms are common to other diseases too, like fever, like dengue virus, viral infection, even malaria to some degree. And so often the early stages are missed. But one of the biggest, at least in the United States, now that we had our first Ebola patient diagnosed there, one of the biggest signs now of possibly being infected with Ebola is travel history. So in other words, did the individual, if the individual is not in West Africa, for instance, did they recently travel from West Africa? And we're in this huge controversy right now in terms of the hospital that's treating the individual, because the individual, when they started feeling sick, went to their local hospital in Texas, and the hospital turned them away. And there's a lot of investigations going on in terms of how that happened. And the last accounting I heard was actually the electronic medical records didn't record the fact that the individual had traveled from West Africa. There have been other stories that the initial treating personnel didn't communicate the information. But whatever happened, that's the time that the individual should have been admitted, but they weren't. They went back home for a couple of days, and at that point is when they potentially infected other individuals. Because the other key about the viral symptoms is once the symptoms emerge, then the person can transmit the virus. So not only is important in terms of the symptoms, being able to determine through the symptoms that the person might be at risk for Ebola, But the biggest issue is then they become infectious. So then when the symptoms emerge, they can become infectious, and then other people can get Ebola from contact with their bodily fluid. So that's a really important point that I think sometimes is missed. So if the person doesn't show symptoms, they're not infectious. So one of the investigations going on is the individual that left. Actually, he came from Liberia. When he flew out of Liberia at that time, he was not showing symptoms. And apparently, when individuals leave Liberia, when they go to the airport to catch a plane, everybody is screened in relationship to whether or not they have a fever. And it was determined actually that this individual at that time did not have a fever, but also in filling out their immigration card, they didn't indicate that they had contact with somebody who had Ebola. And indeed, that individual did. So there's another investigation going on about that. So what is the timeline of symptoms? And again, these days are all ranges. They're not fixed in stone. They're just averages of all the individuals that have been studied so far. So the first symptoms, again, can also appear as early as day 2, right? The first symptoms are headache, fever, fatigue, and muscle soreness. Now, again, those symptoms overlap with a whole lot of other diseases. Now, but if the individual's in West Africa, right, you might want to consider Ebola right away. Number 2, if they traveled from West Africa, you also might want to consider that indeed they may have Ebola. So, the real issue right now, or one of the biggest criteria for determining that somebody might have been infected with Ebola is, are they in West Africa or have they traveled from that area? Day 10. So we're going to see later on the differences and in more detail how the immune system is influenced. But by day 10, so if indeed the individual progresses through those stages, you get a high fever, you get vomiting, you get high blood pressure, you vomit blood, and then this passivity, you become so weakened that basically you just have no initiative, no capacity to even move. I mean, you see pictures of individuals with Ebola and they're just like lying out in the middle of the street because they just can't move. And then day 11, you get further along in the disease, you get bruising, you get brain damage, and this is where you get bleeding from the nose, the mouth, and the eyes, and the anus. Now, this is coming back to the immune system, the picture I showed you earlier, where those endothelial cells are developing these big gaps, and then the blood is just, gushing through those gaps. So day 12, basically, loss of consciousness, seizures, massive internal bleeding, and death. Now, the mortality rate from the case fatality rate is about 50%. So 50% of the individuals who are diagnosed with Ebola actually die. And this is quite a high case fatality rate. So what are the characteristics of the virus? It's a single-stranded RNA negative antisense viral virus. It comes from the class of viruses, the filovirus. It's an envelope virus. It's helical. And a close relative of Ebola is the Marburg virus. And these viruses are very lethal. Marburg also has a very, very high case fatality rate. So we have the capsid of the Ebola virus is helical. It's arranged in a helix inside is the nucleic acid. Remember, the capsid is composed of protein. We have the capsomere, which is I guess an encasing of the nucleic material. And then remember, Ebola is an envelope virus. So outside of that is the actual cell membrane of the host cell. So this is all really important because remember, the Ebola virus actually infects the immune system. So what is the Ebola viral structure? You can see that on the outside we have these protein spikes, and that's actually the antigenic part of the virus, and then around that is, of course, the envelope, and inside of the envelope is the helix and the capsomere. And remember that in viruses we have viral DNA and viral RNA. And in this case, because Ebola is a viral RNA, then we have basically the genetic material is RNA. And it's interesting about these RNA viruses because they don't need to get to the nucleus. They just go ahead and make their viruses in the cytoplasm. But what kind of cell do they infect? They infect macrophages. Macrophages are constantly dividing. So they found themselves a rapidly dividing cell to replicate itself, right? But even if the cell wasn't replicating, the fact that this virus can actually recreate itself in the cytoplasm, right here, so you can see that. And the other interesting thing, these the genetic material is really almost like like the transposons of the bacteria. So in this case, we have a full-length Ebola viral genome, but then we have these parts, right? We have these DNA, or not DNA, but these RNA sections. And basically what they're doing in the cytoplasm is that they're just going ahead and doing protein transcription and translation, but they're not creating cellular protein. They're actually creating viral protein. And then these viruses are being assembled in the cytoplasm, and then they're released. So remember, when they get released, they take along the cell membrane because they're enveloped, and then they go on and then they infect other cells. So this is a picture of RNA viral replication, and it's just a generalized slide that shows all different kinds of RNA replication. So, you know, we have double-stranded RNA replication, and we have single-stranded RNA with the antisense portion of the RNA. And so with the negative strand, what they do is they create the complement, right, which is considered to be the positive strand. And the positive strand is needed to actually transcribe, or I guess if you want to call it, transcribe and translate the proteins, the viral proteins. And then the negative strand hangs around, right? And then that negative strand is what ends up getting incorporated into the capsid. And then the capsid goes on and basically the whole thing is assembled with the RNA inside of the capsid. And then the viral particle is released, and then it can go on and attach and infect other cells. So this is really quite an efficient way to actually replicate itself, right, and propagate itself and cause a much bigger infection from, even just a mild infection, right? You can just have a little contact with bodily fluids from a Ebola patient, for instance, and you could go on and develop a full-blown infection. So one of the reasons why the healthcare workers wear it looks like a spacesuit, right, is because you just only need a little bit of contact, and then, my God, going through this whole process, you can develop a very serious infection. And so one of the reasons for all of the precautions that are being taken is really because Ebola is a very, very infectious virus. So this is another picture of the actual entry of the Ebola virus. So remember back in our generalized discussions that we had about some of these attributes about bacteria, viruses, just generalized concepts that we learned about in the whole course. How do we get an infection? Number one, we have to have entry. We have to have attachment, remember, and then we have to have entry. And so if we have contact with bodily fluids, we get the virus that basically interacts with our own body fluid. The immune system sees the Ebola virus as a foreign particle. What does it do? It launches a inflammatory reaction. These cells migrate. Macrophages are one of the first cells on the scene. And lo and behold, that's the actual cell that the virus penetrates. Okay, so we get, attachment and attachment, right, and penetration. And then we, just by that process, right, then we get the inflammatory reaction, where one of the hallmarks of an inflammatory reaction is that these endothelial cells, I guess dilate, right, and the gaps between them get larger so that other immune cells can travel to the site of injury. But in that process, we actually have the macrophages traveling with the infectious particles, with the infected virus, or with the viruses inside of the cell, and then the viruses can actually get released into the individual's body fluid. And it's this time, right, that individual becomes infectious. So, depending on the type of response we have, there are other parts of the immune system that actually get turned on or suppressed. So in the later stages of the Ebola infection, these endothelial gaps become very, very large. And then, that's when we have these extensive vascular leakage, problems with coagulation, and the whole dysregulation of the cardiovascular system. So there seems to be two different trajectories of individuals with Ebola. So some individuals, as we said, there's about approximate 50% case fatality rate. So the individuals who survive Ebola actually are able to reduce the viral load through actions from the immune system. But individuals who die from Ebola, their immune system is not able to eradicate the infection. So one of the things that is going on is trying to determine what are the cofactors that are required for a more extreme form of the Ebola infection. So in other words, are there other diseases that individuals have when they die from Ebola? Are there other reasons that they may be more susceptible? But remember, one of the criteria that we discussed early on was the effective dose. Remember, so often individuals who have a higher viral dose actually become sicker, right? It makes sense. The more virus you take in, potentially, the more possibility you have for having a severe form of the disease, and the greater the likelihood for mortality. So here's another picture about this interaction between Ebola and these macrophages. So remember, the early form of a macrophage is actually called a monocyte, And when that monocyte becomes infected, it does its job, right? It secretes all these cytokines, but it can be so overly infected that we create a cytokine storm, which serves to further the gap between those endothelial cells. And then we get even dysregulation, we get cytopathic effects, we get death of the macrophages. And then when the macrophages die, whatever capacity they had to take out, to phagocytize the virus is gone. So when you start losing these immune cells, then your capacity to fight the infection is greatly diminished. So here's another picture about the full pathogenic process. So we get infection from the filovirus. Of course, the Ebola virus infects the monocytes. It also, gets taken in by the neutrophils. And remember, the neutrophils also also participate certainly in inflammation. And then we get depletion. So we actually get depletion of the natural killer cells, and those are one of the cells that certainly eradicate the pathogen directly. And then, so, our neutrophils are doing their job by secreting all these cytokines and degranulating and so forth. And we get inflammation. We get, initially a increase in immunity. But then the other part of this is that the actual Ebola virus can serve to actually suppress part of the immune functioning. So it can serve to actually suppress the T cell part of the immune reaction to the Ebola virus, and that can certainly serve to diminish the immune response. So here we show that the Ebola virus can certainly infect the liver cells as well. That leads to organ failure and of course, a hemorrhagic reaction. We can have endothelial leakage. We can then that's very visible in relationship to the bleeding and the rash. And, this can produce a very high viremia, a very high viral reaction. And late in the disease, unfortunately, there's just not much hope. There's a diminished immune reaction. There's an increase in virus. And basically, again, this is a zoonotic infection, and it's very, very severe. So how contagious is Ebola? So actually, in terms of contagiousness, Ebola is not a very contagious disease. Now you might think, well, gosh, why is everybody so worried about it? Well, it's not contagious, but it's very infectious. Okay, so it's not, let's make a note of that. So it's not contagious, but it is very infectious. So in other words, what that means is it's a little more challenging to get Ebola as compared to measles, for instance, but once you get Ebola, it is very infectious. It's very capable of 50% mortality rate. So it's, in that sense, it's a very deadly organism. So in relationship to other diseases, and look at the so-called less contagious diseases. These are blood-borne infections. Airborne, SARS, mumps, and the measles. Now, they have said many times Ebola is not an airborne viral disease, and people are getting afraid that maybe it is, or that there's been some discussions that it may change and become an airborne infection. But right now, that Zaire Ebola virus is not airborne. So how does Ebola transmit between individuals? So the first individual, that patient 0 of West Africa, was thought to have become infected by an animal through a contact with an infected animal. So then you might think, well, how does that occur? Well, there's a practice in West Africa called eating bush meat. So then you might say, what is bush meat? Bush meat is monkey meat. Okay, so they eat bush meat. And so one of the ways in which you can get Ebola is if you consume meat from an animal that has been infected. Now, certainly in the United States, that doesn't happen, but it does happen in West Africa. But, in general, the ways in which you can, I guess, become infected with Ebola is through contact with blood or body fluids, urine, saliva, feces, vomit, and semen. Plus, now they're also saying sweat as well. Okay, so also, though, needles and syringes. So just like with HIV, if you handle an infected needle and it pierces your skin, then you are in danger of becoming infected with Ebola. And then, of course, interaction with infected animals. If you're bitten by an infected animal, for instance, that can certainly transmit Ebola as well. But so now looking at the graphic, you cannot get Ebola through the air, you cannot get it through the water, and you cannot get it through food unless, of course, you eat bushmeat. Now, why are healthcare workers at such a high risk? Well, healthcare workers are the ones who handle bodily fluids. One of the things that happens with Ebola patients is they are excreting a lot of blood because of the disease, a lot of vomit. They're vomiting blood. They just are very, very infectious because the hallmarks of the disease are to excrete a lot of body fluids. So just by touching, body fluids, you can actually, develop Ebola that way. If you, happen to shake, touch bodily fluid and then touch your mouth, my God, that's a certain way that you can become infected. And there's a lot of precautions being taken with healthcare workers because of their likelihood of encountering these body fluids. So you see these individuals with completely covered in these protective suits, very heavy gloves, because of creating a barrier from the body fluid. And they're also likely to use needles and instruments. You can have contaminated instruments. And so therefore you need to take extreme precautions. So a lot of the fear in West Africa, they didn't want to bring people to the hospitals because in their mind that was certain death. So in other words, but you might think about it, well, gosh, what if the individual didn't have Ebola? They had dengue or they had malaria or some of the other diseases that cause these fevers. Then they could potentially get Ebola just by going to the hospital. That's true. But the other reason for bringing the people to the hospital is to prevent them from contacting other individuals who don't have the disease. So that's it's a double-edged sword here, but one of the reasons why they're sending so many people from the United States to these areas is to try and create these field hospitals where they can bring people who are thought to be infected with Ebola and then potentially space them out enough even so that indeed you don't, you have less risk of actually getting the disease from going to those environments. And some of the pictures you see, it's very interesting. You see that the hospital or the treatment area actually has patients that are spaced away from one another out in the open air to prevent this possible contamination. So the treatment right now, there is no vaccine and no treatment, but there is one experimental treatment that they've actually given to the two American healthcare workers who came to the United States from West Africa, and the treatment is called ZMapp. And it's interesting because it's an antibody treatment, but these antibodies have been developed from tobacco plants. And there wasn't that much, it's all experimental, there wasn't that much of the treatment made. And because, well, we tried it on two and we had success, doesn't necessarily mean that indeed it's a perfect treatment. But there's a lot of hope for this treatment. It's actually being accelerated in terms of the clinical trials. So the FDA is putting it on the fast track in terms of drug development. But actually, it's only in phase 1 trials, which is actually pretty early in the clinical trial process. But they're really pushing it. And but the interesting part about it is, number one, it's an antibody treatment, but number two, it's actually coming from the tobacco plant. So, the discussion in scientific circles are, well, it's a good thing we still had some of them around, right? With all the, we're trying to eliminate cigarette smoking, but maybe we'll turn the tobacco plant into a pharmaceutical application. Who knows? But anyway, so there is a lot of development going on with the ZMapp treatment, and there's also some development going on in terms of creating a vaccine. But the vaccine trials are still in animals. And so they're still pretty early stage as well, because before the most recent outbreak, there really wasn't that much going on with Ebola. But aside from these experimental treatment, there are other things that can be done when they're done early, and they can greatly improve the chances of survival. So one is providing intravenous fluids early on, balancing electrolytes. That's one of the reasons. So when you have these gaps in the endothelial cells, you've got to keep making sure that the individual gets enough fluid so that they don't get dehydrated. Maintaining oxygen status and blood pressure, treating other infections if they occur. And really, we saw that one very telling graph. There's kind of two paths that individuals seem to be going on. One is, that lethal immune reaction path, and the other path is one that actually promotes survival. So there's a lot of work continually being done on trying to get a better understanding of this infection. And in fact, there's some, attempts to really, as I talked about earlier, to try and figure out if some certain subsets of individuals are more susceptible than others. So that concludes our lecture portion. Now we have our questions. So the first question we have is, why do some African countries have higher infection and mortality than others? Number one, quality of the healthcare system. Okay, and so for instance, Nigeria is had an outbreak of their own. And remember, if you remember the map, Nigeria is right next door to this area, but they only had one fatality, and they were able to contain and eliminate the infection. It's because of the quality of their healthcare system. What did we see with the three countries, Sierra Leone, Liberia, and Guinea? They didn't have many hospitals. They were poorly served countries by their healthcare system. So that's one of the reasons why the United States is mobilizing, right? It's almost like an army. It's unprecedented, really, an army of healthcare workers. And the army of healthcare workers are designed to contain the infection in those areas. And if we can eradicate the infection there, we will have eliminated the infection in the world. But, with people leaving the area and going to other places, boy, it becomes much more challenging. So the second question is, what are some ways that the Ebola outbreak can be contained? Well, number one, isolation, right? Isolation of suspected patients, right? So why is that? So they don't come into contact with new individuals. So like it's very interesting, the whole discussion going on in the United States. What happened, the breakdown that happened when the hospital didn't originally admit the individual in Texas, right? That individual went back home 2 more days, and apparently they infected a lot more people. Then they came back. So one of the ways, so when the individual is not isolated from other people, they have the capacity to infect many more people. So number 1 is certainly isolation of suspected patients. Number 2 is developing and implementing an effective treatment. If you're able to cure Ebola, then the individuals who are cured certainly will not get Ebola again. But, if we're able to effectively treat the individuals who have or come down with Ebola so that they no longer transmit the disease, and that's certainly a way that we can contain the outbreak. And number 3 is development of an effective vaccine, which is ongoing, but it may be a couple of years before we actually have a vaccine. So what are some ways that risk of infection can be reduced? Number 1, eliminate contact with body fluids of infected individuals, and that's what hospitals are doing now, and they're doing that through protecting the workers, and also through decontamination of the environments. That it's important that if indeed an individual did have the Ebola virus or was infected with the Ebola virus, the environment in which they lived, the contact that they made with other individuals, of course, but with instruments, apparently the apartment where the individual from Liberia was staying when he was in Texas is being completely decontaminated. The ambulance that carried him is being decontaminated. So all of these ways are different ways that we can reduce the risk of infection, but also, do better treatments, better screening methods. So developing better screening methods. So in other words, making sure that individuals leaving that area are truly not infected, and that individual apparently didn't tell the truth on their immigration form, but also when they had their temperature taken, they did not have a fever. So that concludes our lecture on Ebola. Thank you so much for visiting educator.com.